Dr. Mellinghoff is now going to discuss his systems biology work in pathway inhibitors. This falls into the core 2 that Dr. Young talked about. This is attacking the disease with the right drug. So, Dr. Mellinghoff, I have given you control of the screen.
Great, thank you. So I'm Ingo Mellinghoff. I'm at Memorial Sloan Kettering. Thank you for joining. I'm going to just continue what Dr. Young and Dr. Fornari just introduced to you, which is our integrated approach to target specific vulnerabilities in brain tumor, in glioma, in brain tumor cells. And the specific angle that my group with my collaborators is taking is to employ some of the more systems-wide measurements, array-based measurements to identify changes in response to drugs in the tumor cell and the environment. So again, the focus is on the response of the tumor cell to a pharmacologic or genetic intervention, and that is based on the insight from us and others recently that really tumors are adaptive. You can almost think of them as ecosystems, and understanding the response of that system to a pharmacological intervention will teach you escape mechanisms, as you just heard from Dr. Fornari, and also suggest strategies to overcome these. So that's the overlying approach. And so we'll take the leads from the discovery platform, such as you just heard about with regards to EGF receptor variant 3, and then link it to our platform-based measurements of drug response, and then generate hypotheses that can be leveraged in the other cores for clinical development. And I'm going to show you some examples now. So our core, core 2, has several scientific aims. One is focused on the response to inhibitors of mutant IDH. The second one is focused on growth factor pathway inhibitors. So those are different drugs and different genetic vulnerabilities in brain tumor cells. And the third one is a genetic screen that will be looking for potential combination therapies. I'm going to tell you a little bit about these now in more detail. So the first aim focuses on IDH. IDH is a metabolic gene, actually. It stands for isocitrate dehydrogenase. And what makes this metabolic gene so interesting is that it carries a mutation in about 70 to 80% of WHO grade 2 and grade 3 gliomas, and also glioblastomas that have originated from a lower grade tumor. And this metabolic gene, as you might imagine for a protein that's central to the normal metabolism of all of our cells in the body, it regulates multiple biological processes, and not only growth, but also cellular differentiation. And these different aspects of biology appear to be regulated through different effector pathways. And one other reason why we're so interested in IDH as a target in glioma is not only because it's so commonly mutant, that probably tells us something, but also because we have now preclinical data in model systems that blockade of that mutant enzyme with mutant selective inhibitors can be done in vivo, is very well tolerated, and is providing an opportunity, a new target really for the treatment of glioma. And so with regards to isocitrate dehydrogenase, IDH, what we want to do is, and have begun doing, is to dissect the changes in metabolism and gene expression and also cellular differentiation following treatment with mutant IDH inhibitors. And as an extension of this, we are building methodology to measure in vivo these changes in mice and in human in response to IDH inhibition by MR spectroscopy. So those are two major aims we're pursuing. I'm going to show you some examples. So this example here is a flow and complicated cartoon that is a measurement of a metabolite pool, where you're looking at 600 metabolites that are being measured in a cell in response to perturbation of IDH, and it gives you an idea of changes in metabolite abundance. So this was done with a collaborator of ours, Dr. Driggers and Kate Yen at Agios Pharmaceuticals, where you can basically sample the cellular metabolism in a high throughput way in hundreds and thousands of measurements. So this is how we can keep track of metabolism in experimental model systems. The other example or effort I wanted to point out to you is molecular imaging. What you have in front of you here are measurements of the metabolite 2-hydroxyglutarate. So 2-HG is a metabolite that is only made in tumors that have the IDH mutation, and it then accumulates to very high levels, about 100-fold, and it can be detected by MR spectroscopy. And that's remarkable because MR spectroscopy is actually FDA approved for glioma, and we can now leverage this technology specifically for the measurements of this oncometabolite. So it's the ideal non-invasive biomarker of mutant IDH enzymatic activity, and we think it's going to be incredibly useful to measure whether a new drug will get into the brain tumor in patients and is able to turn off the mutant enzyme, because if that's the case, you will see a drop in the abundance of this metabolite. So we're making good progress with this project, and I think it has obvious clinical implication and opportunities. And so the other type of measurement we're collecting, and I'm just going to give you some examples and we can discuss it afterwards in more detail. So these are gene expression arrays where we are looking in this particular experiment at tumors in mice that were treated with either vehicle or a low dose or a higher dose of a mutant selective IDH inhibitor. And you can see, depending on the treatment, the drug dose really, that the mice received, you can see very dramatic changes in gene expression. So when you see a red signal, that's an increase in gene expression, and green is a decrease in this particular image. And so we can, based on these types of measurements, we can identify pathways that are activated or inactivated immediately following targeting of the mutant enzyme. So these are the types of experiments that we're conducting in experimental models of IDH mutation in glioma. And we can use these models, these are xenograft tumors and cell lines, to derive signatures of genes that change in response to mutant IDH blockade, and which we can then measure in human tumors. So that will then link very well with core 3 and 4. So the second project brings you back a little bit to what you heard earlier regarding the epidermal growth factor receptor. And we're taking a very complementary approach to what Dr. Fenari told you. We are interested in this mutant receptor, mutant EGF receptor variant 3. There are also other mutations in the EGF receptor that we're targeting. And what this project is focused on is the observation that the types of mutations that you see in glioblastoma actually respond to different EGF receptor kinase inhibitors than EGFR mutations found in, say, lung cancer. That's a new insight. Specifically, we're interested in what's called type 2 dependent, type 2 EGF receptor kinase inhibitors. They bind to the receptor in a different fashion than the first generation of EGFR kinase inhibitors. And we're actually currently in the midst of clinical trials now, where we give these inhibitors in different dosing schedules. So instead of once a day low dose, we're giving these drugs in a high dose, but intermittently. So we're just completed a clinical trial with alotinib, pulsatile alotinib treatment, where patients take this EGF receptor inhibitor once a week at about 10 times the dose that it was given before. And then we, in some cases, collect the tumor tissue after about a week of treatment. And that will allow us to measure the drug concentration in the tumor tissue and determine whether the pathway is inhibited, and generate really nice hypotheses that we can link back to what Dr. Fenari told you earlier, his lists of genes that he generated, and also resistance mechanisms. So it's closely linked, as you can see here, to both Core 1 and also the clinical trial efforts. And these trials actually are done in collaboration with Tim Clausey and will also with Dr. Young, as you heard earlier. So these are the EGF receptor, the location of these mutations, just to remind you that in glioblastoma, the mutations are in the extracellular domain, and you can see in the Western blot below that these are activating mutations. You see basal phosphorylation in the absence of ligand, and they all are able to promote growth of cells in the absence of growth factor and soft agar. And again, completely different from the lung cancer mutations in the EGF receptor. And this is a differential drug response that I mentioned earlier, where you can see that the glioblastoma cells with EGFR mutation clearly do require EGFR for survival, even though we haven't really leveraged this successfully in the clinic. So this is a major goal for all of our group efforts, is to translate these insights from Dr. Young, Dr. Fenari, myself, Dr. Cavaney, Dr. Michel, into effective EGF receptor therapy. So the trials that are linked to these basic science efforts are outlined here, where you can see that in the red box here, patients take drug for a few days prior to surgery, then they have surgery, and then they go back on to the study medication. So these are different ways that we use to measure pathway inhibition. So I think at this I will stop. The third part are in vitro hairpin screens, which we'll conduct and report to you probably on a separate occasion. Thank you.